Today we will be discussing the major histocompatibility complex, which plays an extremely important role in immune reactions. So, let us try and first understand how this came about. The major role of MHC molecules is in graft rejection. So, basically in terms of skin transplants, in organ transplants, MHC matching is done and it plays a very important role. So, before any transplant, either skin or organ transplants, MHC matching is absolutely required. And why is that required? That is because our body has developed mechanisms by which we reject other people's skin or tissue unless there is a close match between MHC, and this point is something that is going to be discussed in this lecture. The main molecule or molecules that are responsible for graft rejection, organ rejection, tissue rejection are MHC molecules, and these are encoded MHC class 1 and MHC class 2. So, we will need to understand these aspects in greater detail. Okay. The studies on MHC molecules were actually studied as part of transplantation antigens or molecules that are important in transplantations, and subsequently what was shown is that there was a particular loci in the human chromosomes as well as in the mouse chromosomes that were important in rejection of transplants. And often some of these transplants, at least in the mouse, were done in terms of acceptance or rejection of mouse tumors, and that is something that we will be discussing a little bit later. But suffice to say that the MHC molecule is a large cluster of linked genes. It is a very gene dense region in chromosome 6 in humans and chromosome 17 in mice that encode proteins responsible for immune responsiveness. And these, as I mentioned, mainly in terms of transplant rejection, immune responses to pathogens, susceptibility to autoimmune diseases, etc. Now, how is it that the MHC molecules play this role? And that is because you need to think of MHC molecules as essentially peptide receptors. So, they present peptides that are derived from endogenous proteins within our intracellular proteins within cells, and these peptides are present up on the surface of cells, and this MHC molecules presents these peptides, and so the T cells then recognize this complex of MHC plus peptide, and either and in case upon infection some aberrant microbial proteins or peptides are produced, you have a T cell reaction. Alternatively, during tumors, you have tumor specific peptides derived from tumor specific proteins that are expressed on MHC molecules, and you have a T cell response. So, essentially what the MHC molecules do is to present peptides, and that is a readout for things that are going on intracellular within cells. Okay. So, that is what this entire lecture is about, and it all started off with transplantation, because our body has the MHC molecules in our body are recognized by our T cells as self, and so they do not respond against it. However, MHC molecules of other peoples are recognized as non-self, and T cells mount a vigorous response against this. This is a very important concept, and it plays it has ramifications in all sorts of immune responses, and so we will move on and see and discuss this a little bit. Now, as you are probably all aware that in terms of adaptive immunity, there are two types of molecules that are important. One is the humoral response, which is primarily mediated by B cells and immunoglobulins, and the other is the cellular response, in which you have T cells. Now, the T cell receptor recognizes the MHC molecules with peptide, and this is where the importance of MHC comes in, because these MHC molecules are encoded by the major histocompatibility complex. An important point in terms of evolutionary fact is that the adaptive system is absent in jawless fishes and lower organisms. However, they appear to have suddenly arisen in jawed fishes. And so, in jawed fishes you have immunoglobulins, you have the T cell receptor, and you have the MHC products. So, all of a sudden you go from jawless, where you do not have the MHC molecules and the typical B cell receptor and the T cell receptor, to jawed fishes, and suddenly you see this emergence of the B cell receptors, the T cell receptors, and the MHC molecules. And this sudden occurrence of these molecules is known as the Big Bang in immunology. This is again something important for you to sort of remember. The MHC complex, or this loci, as I mentioned, is an extremely gene dense region, and it encodes about 4 million base pairs. So, it's a fairly large part, in terms of the genetic localization, because there are so many genes that it encodes, and several of these genes play an important role. As previously mentioned, it is a set of tightly linked genes that play an important role in transplant and cell mediated immunity, and primarily because they present the peptides to T cells, and you have T cells that can recognize self versus foreign MHC, and so on. Okay, now there are two main types of MHC molecules. One is class 1 and the other is class 2. The structure of these molecules is very distinct. So, MHC class 1 has a single MHC alpha chain with beta 2 microglobulin. So, there is a single alpha, it is non-covalently associated with a beta 2 microglobulin, and the beta 2 microglobulin, the gene encoding beta 2 microglobulin is not encoded in the MHC. In fact, it is encoded somewhere else, but the protein products come together to form the MHC class 1 complex. The MHC class 2 molecule, on the other hand, is composed of the alpha chain and the beta chain. And both the alpha and the beta chain are encoded in the MHC. And this is a structural depiction of MHC class 1 molecules, and you can see this is MHC class 1, and it contains the alpha chain contains 3 domains: alpha 1, alpha 2, alpha 3. And the alpha 3 is linked non-covalently with the beta 2 microglobulin, and the peptide binding groove, or the region where peptide binds, is over here. One of the interesting aspects about MHC molecules, the class 1 and class 2, is that they are highly polymorphic, and the polymorphic residues in case of MHC class 1 are present in this peptide binding region. So, and what polymorphism allows it to bind different sorts of peptides. So, and we will see later that there are two striking features, at least there are two main striking features of MHC molecules. They are polygenic, that means you have different types of MHC class 1, class 2 molecules, and secondly they are polymorphic, that means there are distinct variations or variants of each of these, and this is important because the polymorphisms allow different peptides to bind to it, and so this allows for peptides derived from different proteins to bind to these molecules and to be expressed on the cell surface. This gives an advantage for the T cells to find out what exactly is going on intracellularly. The other MHC molecule is the MHC class 2 molecule, and over here this is composed of the alpha chain and the beta chain, and you can see here the peptide binding group is present over here, and in this case, in the case of MHC class 2, the polymorphic residues are present in the alpha 1 and in the beta 1. And so, this is what forms the polymorphic sites which allow for peptide binding. So, polymorphisms in MHC are very important, and students should be extremely aware of the role of polymorphisms and the advantage of having such polymorphisms. If you remember, the ability to bind the TCRs and the BCRs are variable, and that variable comes from the ability to bind to different segments of the gene and form new molecules. And in case of the BCRs, subsequently you have somatic mutation which drives for selection of higher affinity immunoglobulins. In case of the T cell receptor 2, you have the combination of different genes coming together to form the T cell receptors, and these are diverse and they recognize MHC molecules. In case of MHC molecules, diversity comes in mainly from polymorphism. So, this is an important point to differentiate about the mechanisms responsible for diversity between the B cell receptor, T cell receptors and the MHC molecules, and this is something that students should pay close attention to. Okay, before we go into MHC molecules, I would like to take you into a little bit of historical aspects, because I think it is important for students to understand this. Really, the start of MHC molecules comes in with the World War II, and as you realize that with World War II you had a lot of patients who were suffering from burns, and skin transplants needed to be done. And a certain amount of energy was spent and resources were spent in trying to find ways by which patients would recover much better from skin transplants. And it is over here that one person's work stands out, and that is Peter Medaver. Peter Medaver worked on this aspect because that was a pressing need at that point. Sometimes necessity is the driving force behind discoveries and inventions. And what he observed was that upon skin transplantation, very often the rapidity with which the transplant was rejected was directly proportional to the amount of these small cells that would come around the transplant. And he did not know at that time what these small cells were, but he thought the cellular immune response was responsible for it, and these were important for rejection. And based on his observations, he was a very astute scientist. He came up with what is known as the laws of transplantation. The important aspect was, he found that if the skin transplants were done with relatives, the chances of rejection were less. So there was some matching that could be done, and that helped in improving the chances of skin acceptance. Subsequently, and because of his pioneering studies, he was awarded the Nobel Prize along with Macfarlane Burnet, and who had propounded the clonal selection hypothesis, and both of them received the Nobel Prize in 1960. And so Medaver's contributions in terms of transplantation laws and his observation about why is it that we accept some grafts versus we do not accept some grafts is really important. A critical observation that helped all this came from Ray Owen, who a lot of people may not have heard about. Ray Owen had made a striking observation. What he found is that in cattle, that is in cows, if the cattle gave birth to dizygotic twins and who shared the same circulatory system in utero, they did not respond to each other's antigens, which in other words these cattle these twins were tolerized to each other's antigens. Usually that was not the case, but in case of the dizygotic cattle who shared the same circulatory system, this happened. And this was because of the role of immune tolerance. So one gets tolerized very early on during development. So if our immune system sees something very early on, then you get tolerized, and this lasts a lifetime. And Ray Owen's work was very important for Peter Medaver to come together to put together his laws of transplantation, and this is very important in understanding this aspect. Jean Dosset, he found antibodies against leukocyte antigens in the blood of transfusion patients. So people who had got transfusions, their immune system react to it, make the antibodies against different MHC, and he used these as reagents, and this was the first demonstration of anti-HLA antibodies, and these reagents were extremely important because what happened at that time, there were a lot of studies that were going on in mice and all, and Dosset's studies were important because they were done on humans. So we had the first ideas about HLA molecules at the protein level, and his reagents were very extremely important in terms of HLA typing. One of the first starts of HLA typing came from Dosset's studies. Okay, as mentioned, a lot of the studies with HLA have to do with mice. And what are the advantages of using mice for research? First is they are easy to handle, they have a low gestation, so about within 4 weeks you have the litters, and the litters are fairly large, so you have several of them, so they breed well. You can also time the pregnancies too, so this is important for developmental studies, and it's more relevant as they are closer to studies on mice are obviously more relevant because they are closer to humans compared to Drosophila or yeast like Cerevisiae. And in this stage, it's important to understand about the use of inbred strains of mice. What are inbred strains of mice? These are genetically homogeneous strains of mice, and they have been obtained by brother-sister matings for about almost 20 generations. What this does is that this allows the genome to be almost identical in mice of a particular strain. And why is that important? This is important in terms of scientific experiments and reproducibility of results. So, for example, if some experiment is done with a particular inbred strain of mice in Bangalore, the same results should be mimicked by that strain in Delhi or maybe abroad in the US or Great Britain, wherever studies are done. So, this allows for reproducibility, so there are no differences within the genome of these mice. So, inbred strains of mice are critical for the use, and this is especially true in case of MHC, given that the MHC is a polygenic as well as polymorphic. Okay. Now, with respect to the using mice as a model, there are several people involved. Perhaps the person who we all need to thank is Abby Lathrop, this retired school teacher. She bred and supplied mice for research. In fact, several of the mice strains that we have in our or that we use in our laboratories have come from her farm. For example, C57 Black 6 is actually a female number 57. Now, remember, the strain of mice was derived from Abby Lathrop's farm, and then subsequently inbred strains were developed much later, but the line came originally from there, and that was worked on. And Abby Lathrop supplied mice from her farm to William Castle, who was the head of the Bussey Institute at Harvard University. William Castle, or Bill Castle, started work on using mouse as a model system. He thought it was more relevant to use mice, as was mentioned previously, and so he started serious work on mouse genetics, mouse biology at the Bussey Institute. And what is really important about it is that several scientists, for example, Clarence Little, George Snell, who made major contributions in mouse genetics, immunology, etc., were trained at the Bussey Institute. And one of the members, Clarence Little, developed the inbred strain of mice. For example, C57 Black 6, BALB-C, which is Bag's albino. Bag was a scientist, and it's albino because it's white in color. And more importantly, Clarence Little founded the Jackson Laboratory in Bar Harbor, Maine. Those students who are interested in mice research are strongly urged to look up Jackson Laboratory in Bar Harbor and read up on the role of this laboratory in mouse genetic studies. The Jackson lab distributes mice strains to different researchers all over the world. They have a large collection of mouse mutants, especially some of the ones like FAS, FASL, leptin, leptin receptor, etc., were all generated in the Jackson lab and have been very important in finding the roles of these these molecules. For example, FAS and FASL are important in apoptosis and in the immune system because it's a way of getting rid of cells that are activated and cells that we don't want. Mutations in FAS, FASL result in severe problems, and this is something that we will be looking or we will be studying in subsequent classes. Leptin and leptin receptor are important in terms of satiety or after food, the feeling of satisfaction, and weight control. So, this is again, these were all discovered at the Jackson laboratory. They have also played a leadership role in sequencing of the mouse genome, and therefore, the Jackson Lab's contribution to mouse genetics is really critical. It was also the place where George Snell worked and discovered the H2 or the mouse MHC. And how did George Snell do it? So, the readout that he used was the ability to reject tumors in mice. So the tumors in mice that were self were accepted as self and they were not rejected, whereas once they were implanted in mice with a different MHC, they were promptly rejected. And so that was the basis of the assay by which H2 was discovered. And George Snell collaborated with his friend Peter Gorer and discovered this complex. And H2, why is it 2? H2 was the second genetic loci involved in control of expression of antigens important for transplantation that Gorer had had found. And so the collaboration proved to be extremely useful. George Snell is mainly known for his development of congenic mice. What he developed were strains of mice that were genetically identical except for one loci. And in this case was MHC, because again he used the breedings and the assay system using the tumor acceptance rejection system to develop these congenic lines. So you can what this told, that there was a particular loci that was controlling the ability to accept or reject transplants, and this loci turned out to be the H2 or the MHC complex. And so this was critical in our understanding of the MHC, and along with Baruch Benacerraf and Dorset, they were awarded the Nobel Prize in 1980. Baruch Benacerraf's contributions are mainly in our understanding of the role of IR genes, and IR stands immune response genes, that is the ability to respond to an antigen. And the IR genes were subsequently found to be MHC class 2. And you need to understand the MHC class 2 is important because MHC class 2 presents peptides to CD4 positive cells, and CD4 positive cells produce cytokines which activate the B cell receptor. So what was found is the IR genes control the amount of antibodies that were being produced to certain antigens. So if you take the same antigen and give it to a mice containing different H2, you got different responses, and these responses were determined by the IR loci, and IR is nothing but MHC class 2 as was subsequently found. And I hope you are able to understand this relationship, because the antigens were processed and presented on MHC class 2. Now, depending on the polygenic and polymorphisms in MHC class 2, these different peptides were presented and would activate T cell responses or CD4 T cell responses. This differential CD4 response would translate to the amount of different antibodies or different titers of antibodies produced. So you had some strains that were high responders and some strains that were low responders, and this was determined by MHC class 2, and consequently you got a differential response, and that was the IR loci. Subsequently, what was found, there were some critical experiments done by Rolf Zinkernagel and Peter Doherty, who showed it's actually the MHC molecules plus antigen that is responsible for recognition by the T cell receptor, and this is known as the altered MHC. And for this they got the 1996 Nobel Prize. So it's the self MHC that, together with a peptide from a pathogen and this altered self MHC, presents or is now recognized by T cells that is responsible for T cell activation, and for improving our understanding on MHC restriction, they received the Nobel Prize. In all these great experiments and contributions of MHC molecules, there has also been a problem. In 1972, you had the case of W.T. Summerling, who showed that skin transplants that were very difficult to do could be done between strains of mice, and what he had actually done was had used ink to fraud his data, and that was not something nice. But I feel students should be aware of these, and it's such a famous case that that's why I have included it in my lectures. Okay, so the MHC molecule In case of humans, MHC class 1, you have the HLA A, B and C, and in case of mouse, you have the H2, K, D and L. And remember, these are the single alpha chains, and each of these alphas are in contact with beta 2 microglobulin. The MHC class 2 molecules, in case of humans, you have the HLA DR, DP and DQ, whereas in the mouse you have IA and IE. Now, in other animals also there are MHC molecules. They are not as well studied as mouse and humans, but one should be aware of it. So, in rat, for example, it is known as the rat RT1 molecule. Okay. So, the MHC class 1 molecule is present on all nucleated cells, and they are recognized by CD8 positive cells, and so the MHC class 1 is mainly responsible for the cytotoxic T cell response. The MHC class 2 is expressed only on antigen presenting cells, or primarily on antigen presenting cells, and it is recognized by CD4 response, and this is where the IR loci part comes in, and that is something that I discussed in the previous slides. Okay, and so the MHC class 1 presents antigen to CD8 positive, and which leads into generation of cytotoxic T cells, which is important in elimination of tumor or infected hosts. And in MHC class 2 is presenting of antigens to helper T cells, which secrete cytokines, and these cytokines in turn influence the B cell response. An important aspect about the binding of these different peptides are the role of anchor residues. So, if you look at different MHC molecules, and because of polymorphisms, you have different polymorphisms, but there are some residues that are fixed. So, especially in this you have this position of the peptide is fixed, and these are known as anchor residues. So, in position 5 in the binding of H2K of B, and the B is refers to the particular allele. You have either tyrosine or phenylalanine in position 5 and leucine at position 8. And in case of IA of B, which is MHC class 2, you have position 3 asparagine, and in position 9 you have proline. So, you have these anchor residues, and these anchor residues, what they do is they help the binding to MHC molecules much better. So, even though you can have variations in these in the amino acids in these other regions, you have these anchor regions which sort of ensures that the peptide binds strongly to MHC molecules. So, anchor molecules are important in that respect. Now, the MHC loci, you have different types of genes. Now, I have been talking mainly about class 1 and class 2 molecules in the MHC, but I do not want to give the impression that these are the only genes that are present in the MHC. So, you have certainly you have the genes encoding MHC class 1 and class 2. You have genes that are important in antigen processing, and this is something that we will be seeing in the next two lectures, both for MHC class 1 and class 2. The genes important for antigen processing are present also in the MHC. Now, why do you think that would be? You know, one of the reasons that have come up is that both the structural genes as well as the genes important for antigen processing and antigen presentation, if they are together as part of the complex, then they would be sort of carried together down, and that could be one possible reason. But there are, you know, that is certainly one reason, but beta 2 microglobulin, which is so important, is not part of the MHC. So, you have both sides of the argument, but nevertheless the MHC loci contains different sorts of genes. You have genes encoding complement proteins and tumor necrosis factor alpha, which is important in the cytokine response. You also have genes encoding non-immune functions. Now, this is a depiction of the MHC loci. This is the human HLA, and you can see DP. DP is the MHC class 2, DP, DQ, DR. Now, in between DP and DQ, you can see DM, proteasome genes and TAP1, and this is something that we will be discussing in the next classes. I just want you to focus on the proteasome genes and TAP1, which are important antigen processing and presentation genes in case of MHC class 1, and whereas the DM is important in antigen processing presentation in case of class 2. So DP, DQ, DR are important parts in the structural genes important in MHC class 2, and the corresponding genes are shown over here as IA and IE, which are present over here in case of mouse. And what has happened is in human, the MHC class 2 loci is very straightforward. You have the MHC class 2 loci, then followed by the class 3, which encodes for complement and lymphotoxin, tumor and necrosis factor, and then you have the MHC class 1, where you have HLA, B, C, A that are present, and along with it you also have class 1 like genes, pseudogenes, etc. Now, in case of the mouse, you have the MHC class 2 loci, you have the MHC class 3, and you have MHC class 1. Now, here D and L are present over here, the H2D and L. However, H2K is actually centromeric, it's present over here, and so you can see that there are some differences between the organization of the HLA and the H2 complexes, but you can see that it's a very gene dense region. You have different sorts of genes being encoded by these loci, and very and several of them are very important for the immune response. Okay. So, one way by which we can differentiate these different genes and the roles of these genes, and the analogy that I use is, you have one world, but that world is divided up into continents, and continents within a single continent you have countries, then you have districts, and then you have villages. So, there is a different way by which you can differentiate this world, which is composed which is diverse and composed of different different constituents. Similarly, in case of proteins, you have superfamilies, you have transcription factors, you have immunoglobulins. Now, the immunoglobulin family is further divided into real immunoglobulins, you have TCRs, you have MHC molecules, you have adhesion, so on. Now, the MHC molecules can be further distinguished into classical and whereas non-classical MHC. And in the classical ones are the ones that are primarily responsible for transplant rejection. You have MHC class 1 and class 2. Now, within class 1, you have HLA, HLA A, B, C, and these are the different polygenic forms, or in case of mouse you have K, D and L. And now, within each of these you have different variants, and these are responsible for polymorphisms, and I showed you where the polymorphisms lie, and they lie in and around the peptide binding region, which is an important aspect. So, you have K, which you have different variants of K. So, you have K of D, K of B, so on, and you have different alleles of, in case of HLA molecules, you have HLA B2701, HLA B2705. These are different variants, and these are important because these variants give us the ability to bind different types of peptides, and they are important in terms of the immune responses and transplant rejections also. So, this is something we need to keep in mind. Some of the key features of MHC molecules, overall these are the important features that we need to understand. Their role is in binding and presenting peptides to T cells. This is their primary role, and what is important about the fact that about the peptide binding is that peptide binding stabilizes cell surface MHC molecules. So, it is very important for peptides to bind to them because it stabilizes their structure and stability. Second is their genetic localization, and genetic localization, they are present in this loci known as the MHC. It is HLA in case of humans and H2 in case of mouse. The third important point is that these molecules are interferon gamma inducible or inducible by certain cytokines. Now, why would that be? Now, during an immune reaction, you have immune responses. So, more the MHC that is produced, more the chances that a T cell will recognize a cognate MHC TCR complex to which it can respond. So, that is an important aspect that one needs to keep in mind. The most important aspect about MHC molecules is that MHC molecules are polygenic as well as polymorphic. You have different forms of MHC molecules K, D and L, and within K you have K of B, K of D, K of F, so on. Now, MHC molecules, as I previously mentioned, you have two different types. You have the classical and the non-classical, and MHC class 1 you have the heavy chain, which is 45 kDa, which is linked non-covalently with beta 2 microglobulin, which is the 12 kDa. MHC class 1 is expressed on all nucleated cells, and the peptide it binds peptides which are about 8 to 10 amino acid long. So, they are much smaller. MHC class 2 you have the alpha chain and the beta chain. The alpha is about 35 kDa, the beta is about 28 kDa. Now, as mentioned previously, MHC class 2 expression is present on primarily antigen presenting cells. So, you have B cells, macrophages, dendritic cells, Langerhans cells in the skin, and the peptide binding over here is much longer. So, it is about 14 to 20 kDa, and the responder T cells are the CD4 response. So, CD8s are primarily recognize MHC class 1, CD4s primarily recognize MHC class 2, and this is where the IR genes come into play. I also mentioned the differences between the H2 and HLA, and it is important because I showed where K is centromeric to the H2K is centromeric to the class 2 region. There are two MB regions. MB is important in MHC class 2 antigen processing and presentation. In H2 you have two of them, whereas you have a single one in HLA. There is no DP equivalent in the mouse, so the DQ equivalent is IA and the DR equivalent is IE, but there is no DP equivalent. H2L is deleted in most mouse strains except for some haplotypes which are shown over here, and these are those variant the haplotypes are the H2D, H2Q or H2R. So, that represents the variant form H2D. Now, HLA G and HLA E equivalents are missing in H2, but Q and TL are present. Now, both Q and TL or thymic leukemic antigens are non-classical MHC molecules. And coming to non-classical molecules, as I mentioned, the classical ones are the MHC class 1 and class 2. And these are the ones that are really important for transplant or organ rejection. And then whereas the non-classical MHC molecules do not play a major role in transplants. They have a low cell surface expression, they have a restricted tissue distribution, which means they play a role only in certain situations, and they are not as polymorphic as the classical MHC molecules. But their roles for some of them have been shown. So, for example, in mouse you have QA, QA1, QA2, and some of these bind to NK receptors. And the thing that one needs to understand is the NK receptors also recognize MHC molecules, and in case the NK receptor sees the MHC molecules, that inhibits their lysis. However, if the MHC molecule is not present, then it will go ahead and kill. You have certain NK receptors that are responsible for this, and so QA is one of the ligands for NK receptor that is known, and that has some role to play in regulation of immune responses. Now, M3 is a non-classical MHC molecule, and what is interesting is that it binds formulated peptides. Now, where are formulated peptides produced? Formulated peptides are produced primarily in the mitochondria or by bacteria. So, by recognizing formulated peptides, what happens is during a bacterial infection, you have presentation of formulated peptides by M3, which may be important in it. So, it has a very specialized function. Human HLA F and you have E as mentioned. Now, E as mentioned as has been shown to bind to the NK receptor CD94 NKG2, and I discussed the roles of this relationship between binding of NK receptors with non-classical MHC molecules and their role in modulating or regulating immune responses. A very interesting and well studied non-classical MHC molecule, perhaps the most famous non-classical MHC molecule, is HLA-G. Now, HLA-G is expressed in placenta, and it may be important in protecting the fetus because it plays an important role in lowering cytotoxic T cell lymphocyte and NK responses. HLA-G is very important because it may be a mechanism by which the fetus is protected, and you can understand the situation that when a mother is carrying a child, the child has a different MHC. So, it is natural for the mother's immune reaction to sort of react against the child's antigens. But however, this does not happen. There are some protective mechanisms that the fetus has by which it protects against the immune response, and it is possible that molecules like HLA-G are playing this important role. And in fact, the expression of HLA-G is that it is expressed primarily in the placenta, and so it might really be important in protecting the fetus. So the non-classical MHC molecules have specialized roles. So it would be interesting to find what are these roles, but the dominant response of MHC molecules comes from classical MHC molecules, which are responsible for organ and transplant rejections. Now, MHC molecules are also involved in other responses, and the one that we will be discussing is this relationship between MHC molecules and disease. So, there is a very strong association with a particular type of HLA molecule, which is the HLA-B27 and spondylitis. So, those people who have HLA-B27 are more prone to this particular disease. And studies have also shown relationship between certain HLA molecules, here in this case DQ beta, and insulin dependent diabetes mellitus. What has been shown is the position 7 is important. So, if you have a valine or a serine, there is an increased chance of having insulin dependent diabetes. However, in this case, if you have in position 7 aspartate, then there is a decrease in diabetes. Now, MHC molecules are important for resistance to pathogens, and this particular MHC molecule HLA-B53 is important for resistance to malaria in Africa. So, these studies have been done. What was shown is if you look at the survivors from endemic areas, and if there is a high preponderance of a particular HLA, then there is a link made between this particular HLA and perhaps resistance. But how exactly this occurs needs to be studied much further. Again, there is a link between HLA, individuals having HLA-A02 and there is those individuals have low incidence of infections with HTLV. Now, cheetahs are extremely sensitive to viral infections and have limited MHC polymorphisms, and it is possible that they are highly susceptible because of their limited MHC polymorphisms. And one of the possibilities is that they have come from a small founder stock, which means that the initial stock that the cheetahs came for were small in number, and then they bred and increased their numbers. But since they came from a small founder stock, their MHC diversity is limited, and perhaps that makes them highly susceptible to viral infections. An important aspect that we need to study, especially with respect to host pathogen interactions, is if the host comes up with molecules, the pathogen also tries to come up with molecules that will evade, and this is seen very well in the interrelationship between MHC and antiviral responses. So, in fact, you have certain viruses like adenovirus E319K, which binds and retains MHC molecules in the ER. You have cytomegalovirus which encodes molecules US2, US11, US3 that targets MHC molecules for destructions. Now, I want students to sort of think a little bit about as to why would viruses encode molecules that target MHC molecules. And it is possible that they do so because if they target MHC molecules, then the presentation of the viral proteins will be hindered, and as a result of which the immune response, or at least the CD8 response, will be less, and that gives them a sort of an advantage. So, you need to think of it in those terms, that you have the struggle between host and pathogen, and both are trying to outwit each other. So, the host has come up with different sorts of mechanisms, and the pathogen is trying to find ways by which they target. And one of the ways by which they target is the pathogen targets is by encoding proteins that target MHC molecules, which tells you about the importance of MHC molecules in pathogen recognition. Okay, now what I would like to do is to go over some terms that are important for MHC studies. So, for example, the first term that we will try and understand is codominant. and we will understand it from the point of view of this example. So, let us say you have a parent which has 2 alleles. Remember, we are 2n, which means we have one from the father, one from the mother, and that is what is shown over here. You have A and A, and you have another person which is B and B. So, and now you have a F1 AB. I want you to answer the following question: will AB accept graphs from AA or BB? And the answer to that is AB will accept graphs because from AA or BB, because the A antigen is present in this F1 individual. So, it will see A as well as B as self, and therefore, will not mount a reaction against it. On the other hand, will AA accept graphs from AB? Not likely, because the B is not present in AA and will be seen as foreign, and therefore, AA will not accept graphs from AB. Similarly, in case of BB, A is not present, and so therefore, A will be seen as foreign and it will not accept graphs. So, it is important to understand this concept of graft with respect to MHC. And the fact is, since we express MHC molecules one from our father, one from our mother, both are codominantly expressed, and that is the importance of codominance. Okay. So, our MHC molecules we are expressing both from our father's side as well as from our mother's side. So, in terms of MHC class 1, in terms of A, B and C, there are 6 MHC class 1 molecules being expressed, 3 from our father, 3 from our mother. Okay. And in case of MHC class 2, you have DP, DQ, DR, again 6 types of molecules that are present, 3 from our father, 3 from our mother. So, we need to understand it from that point of view, and that is why MHC molecules are polygenic and they are codominantly expressed. That means, both the fathers as well as the mothers MHC molecules are being presented. Okay. So, we will now go to the second term, which is MLR or mixed lymphocyte reaction. Now, in this case, we will again use the analogy of AA and BB cells, and now if you want to check for whether AA will respond to BB, what can be done is you can do it 2 ways. One is you can put AA and BB cells together, and what will happen is, especially if you take peripheral blood lymphocytes and put them together, the T cells will recognize the MHC molecules. So, AA T cells will recognize MHC molecules and BB will mount a vigorous reaction. The BB T cells will recognize the AA MHC molecules and will mount a vigorous reaction. So, both you have both types of interactions over here. So, this is it's a 2-way MLR. So, you do not know who is responding against whom. So, in order to understand responses, what can be done is BB cells can be treated with mitomycin or irradiated so that they do not proliferate. So, the cells will be fine, but they will not be able to proliferate, or they will be able to express MHC class 1 and class 2 molecules. So, if you see this, you will get a AA anti-BB response, because these cells are not fixed or treated, and so the T cells over here will recognize the BB MHC molecules and will generate a AA anti-BB response. This is called a mixed lymphocyte reaction. So, if you want to check for MHC responses, what can be done is you can take from blood from the donor as well as the recipient, and you can do these sorts of responses, because it will give you an indication of the response that is seen. If you see a vigorous response, it is unlikely that the transplant would be accepted. You have now finer studies by which, following MLR studies, you could do MHC matching studies by looking at specific expression of specific MHC molecules on the surface using specific antibodies. Further on, further sequencing studies can be done to look at the extent of match, or especially of some of the major antigens, which is HLA-A, B and DP-DQ. So those are sorts of things that are important for transplantation, and this is important that students recognize this. But before doing finer studies, a simple MLR is good enough to tell you about the vigorousness of the reaction of the anti-MHC response. And part of this is because our T cells, about 10% of our T cells you have T cell receptors that recognizes allo-MHC, which is MHC different from self, and so they spontaneously recognize against it, and that is why you have this vigorous T cell response against non-self MHC molecules. The second one is GVH and MHC matching. GVH stands for graft versus host reaction and HLA matching. So, what happens is after HLA matching, once the donor cells are injected in, the graft now recognizes, in case it is possible that if the T cells from the graft now recognize the host cell as foreign and mount a reaction. So, that is what is known as a graft versus host reaction, and that is because there are some T cells present in the graft which have been injected, and these cells are now recognizing the host and they are generating this response. You do not want that. You want to make sure that the T cells from the graft are removed, because otherwise it results in what is known as GVH. Now, despite the fact that you have, as I mentioned, the major histocompatibility complex present, which is responsible for organ and tissue transplantations, you have what is known as minor. So, let me give you an example to try and explain this. So, let us say you have two inbred strains of mice. You have an inbred strain of mice. So, in theory, the T cells from the inbred strain should not recognize MHC molecules against another of the same species, of the same inbred mouse strain, but sometimes this happens, and this happens because of the presence of minor histocompatibility antigen. That means the T cells over here have not been tolerized to some antigens, and what are these antigens? So, one would be a male specific antigen, and this could be this difference between male and females. Even though they are inbred, there are some differences in males and females, because the males have proteins that are encoded by the Y chromosome, and so that may be responsible for it, and you have some of these, or you have some others like mitochondrial antigens which are present. So, these are minor histocompatibility antigens. So, again the response is very small, it is delayed, it takes a much longer time, but these are minor histocompatibility antigens and these need to be differentiated from the major histocompatibility complex antigen, which are the dominant players, and the minor histocompatibility antigens are usually peptides, for example, that are present or encoded by proteins in the Y chromosome, or mitochondrial antigens, and now if you remember, the mitochondria is transmitted primarily from the mother. Okay, I am going to discuss now an aspect, a newer aspect or newer facet of the role of MHC, and that has to do with selection of mates. And this is because in a way what was an interesting experiment was done, and what was found is that MHC, that mice with dissimilar MHC prefer to mate with each other compared to the ones with similar MHC. So, this gave rise to the notion that mate selection, that MHC is also important for mate selection, and how does this occur? And perhaps body odors play an important role in this. Now, what was shown is in humans that you have body odors, and this is primarily because the part of the odors is generated by peptides that is bound on MHC molecules, and these may be responsible, and this is an important, and this drives sexual selection. This is important because if you have dissimilar MHCs, you have now, if you remember, you have polygenic MHC, you have more polymorphisms, and greater the diversity, it is perhaps better to mount immune responses to pathogens. So, perhaps nature has selected for the fact that you try and find partners that are of a different MHC, and that helps in generating a better immune response. And in order to do that, body odors play an important role in maybe helping this out, but the mechanisms behind the body odors are perhaps peptides that are bound to MHC molecules. So, now remember, most of the peptides that are bound to MHC molecules are derived from self MHC are derived from self proteins. Majority of the peptides that are bound to MHC molecules are derived from self proteins, because these are degraded and the peptides are there and are presented on MHC molecules. And so, this is an intriguing intriguing thought and idea, and this is a new facet to the role of MHC molecules, and I thought it's important for you all to sort of understand and appreciate the role of MHC in this process. I will now briefly summarize the lecture. So we had first discovered what is the MHC. MHC is this certain loci, is this particular loci that's present in organisms, and the ones that are maximally studied are humans and mice. And in humans it's known as HLA, and in mice it's known as H2. You have different genes in there, and that primarily dictates organ transplantation. But how it does it? It's actually it does so by the production of classical MHC molecules, class 1 and class 2 molecules, which present peptides and which are important in it. Now, why is it that you have a vigorous T cell reaction against it? Because during selection, and this is where Ray Owen's discovery turns out to be so important, and that was caught up by Peter Medaber, who realized that we become tolerized during development and early on during development. We discussed the brief history about MHC. I discussed about the role of Medaber, Ray Owens, John Dosset, and then the incredible history on mouse biology and mouse research by different workers, Bill Castle, Clarence Little, George Snell. It is then finally coming down to Zinkernagel and Doherty, all extremely classical studies that students should read up more on and get excited about. We discussed the differences between HLA and H2. Remember, in H2 the K is more centromeric. There are differences in the MB genes. You have 2 MB genes in H2, single in HLA. There are differences in the non-classical genes. The most important aspect, of course, is the MHC class 1 and class 2 molecules is their expression and function. MHC class 1 is expressed on virtually all nucleated cells, MHC class 1 on antigen presenting cells. The peptides that bind to MHC class 1 are smaller, about 8 to 10, and in MHC class 2 they are much larger. MHC class 1 stimulates the CD8 response, MHC class 2 stimulates the CD4 response. We also discussed about the role of non-classical MHC molecules. These do not play a dominant role in tissue transplants, and the most famous non-classical MHC molecule is, of course, HLA-G. There are some common features about MHC molecules. They are present, their genetic localization, their induction by interferon gamma and certain inflammatory cytokines. So, the expression is increased, and so more the chances of recognition of MHC peptide complexes by T cell receptors, and the fact that they are polygenic, different genes, and polymorphic, and this allows for greater diversity. So, more the chances of peptides binding and being recognized. We also went over some examples of MHC molecules and immune responses in terms of pathogen responses, and this you can see in terms of population based studies, and this is also important in terms of vaccination, regimes and association of MHC molecules with autoimmunity. Finally, we also I brought forth this important point of MHC and mate selection and the role of body odor. I hope overall you have understood this aspect about MHC, and clearly MHC is important in terms of immune responses, especially in terms of transplants, and immune response to pathogens, autoimmunity, so on. But the overriding feature is really transplant rejections, and especially its role in acceptance or rejection of grafts. That is how MHC molecules started off with, and that is what we are still following in terms of looking for acceptance or rejection of organ and tissue donors. But to understand T cell biology, you need to understand MHC molecules, and we will be in the next class studying more about the mechanisms by which peptides are generated for MHC class 1 and MHC class 2 and how they are presented. Thank you.